Okay, Filza. So today we are going to talk about uh, like I have started with you photosynthesis and respiration as a concept that we
Yes.
discussed. It's basic, like what is photosynthesis and how it is different from respiration, right?
Yes.
So um, if I just follow Alberta curriculum, so definitely few of the things that you must know as a prerequisite before going
Hmm.
to talk about these complex concepts.
Okay.
It's a very basic knowledge, where we just need to understand different structures which are present in cell. So the knowledge you must have before going to talk about the photosynthesis and respiration, um, the first important concept is we must be able to understand the different type of subcellular structures which are present in animals are and plant as well, like animal cell and plant cell. How both of these types of cells are different from each other, this is what we are trying to know. So let's try to understand this structure first, and then we'll go to talk about the concept of photosynthesis. Um, like, what is the difference between photosynthesis and respiration? What not? What is this? I'm trying to, just a moment. It's not copying. Copy. I guess board is not working. It's taking too much time. Okay. Can you see this? Here is a structure in front of you. Is this visible, Filza?
Yes, it is.
Let's try to work on this. So if I can just
Mhm.
understand, like this is one side, you can see it's animal cell.
The second one is animal cell.
This one,
Yeah.
are you sure?
No, that one is plant, the other one's animal.
Yeah, this one is.
Animal, the other one's plant.
God, what's happening? Are you doing something from your side? What's going on here? Uh,
Huh?
s- Sorry, actually, it's not working, so I'm just checking what's the problem. Now it's no, again, it's not. Okay, just a moment. I just want to erase it from here. Might be erase it. It's diagram, so that's why. Okay, so it's as you said. I'm not able to write. Just a moment. Not scrolling. Okay, right click. What do you mean by right click? Okay, animals. No, should not be like that. Sorry, Filza, just a moment, please. I'm just trying to figure out where is the problem. Actually, I'm using my tabs, so never happened before, but I don't know why it is so. Let me check now. It's animal cell, as you said, and
Yes. The
this
other
one.
one is plant cell.
Okay. There was a problem here in this. So this one is, as you said, it's a plant cell. Okay. So this is a general identification. But let's start with the A. Can we identify what this structure is in animal cell?
Which one?
Sorry. A.
I know this one. Give me a second though.
Yeah, please.
I want to say something, but then I feel like I'll say it wrong.
No, it's okay. Well, I mean, that's I'm saying, Filiza, like,
Wouldn't that be considered the nucleus though?
yes. So why you're saying that? Why you doubtful about this?
Because
And
I was confused between
no,
cytoplasm and the
that's why
nucleus.
you're taking class with me, Filiza. That's I'm here to remove your confusion. So whatever that should be in your mind, so please, you should participate in the class. So this would help me to understand where you are standing, and based on that, I will help you as well. So
Okay.
we'll try to work on that. So yes, you're correct. So it's nucleus, and there's another structure B. What is this structure? Let's try to figure out.
B
It's
wouldn't it be the wall?
this is animal cell. So animal cell does not have a cell wall, but it has another structure. That's what we call cell membrane. Okay, because in case of plant cell, there's additional layer or wall that's called cell wall, right? But that is not present in case of animal cell. Are you getting? So
Yes.
it starts with the cell membrane. Okay.
Okay.
And after B, what's next? A, B, with C, with D, D, A, B. Oh, yeah, C is here. Okay, let's try to understand what this structure is. This is in plant cell.
Okay.
Uh, this green structure.
The
Yes,
green
Filza. Just
structure.
guess. Just one guess. Any guess. I'm just asking for any guess.
Chloroplast?
Wow, how do you know?
I guessed, honestly.
Okay. Yeah, it's chloroplast. Good. Yeah, exactly. This is chloroplast, and this is a green part of it. And it's a very clear structure, like, and this is a structure because of this photosynthesis is possible in plant cells. Okay.
Yeah.
So again, there is another structure, A. What is the structure here? Is this not very similar to this structure?
Which one?
Like this side, on animal side, this
Yeah,
structure.
the orange thing.
So this is quite same. So what should I
Yeah.
say? What is this structure?
Uh,
Can I say it's nucleus?
no, but which one? The one that you circled?
Like this one. Just like because we have
Oh, the
structure,
purple one.
this is in animal cell and this one is in plant cell.
So if we have a nucleus, then it would be the nucleo something.
Nucleolus. Are you talking about nucleolus?
Yeah, because that's the only part that comes to mind.
Um, Nucleolus should be here, like in the middle dark portion. But the point is, in this case, this A is nucleus. You know, this is the nucleus of animal cell, and this is the nucleus of plant cell. There's no difference. Like both of the structures are quite same. So there is no need to be confused here. Nucleolus is the dark structure, which is present here. Like this is also present in both of these cases. What's this color? It's a purple color. I like that. What's this color? You know better.
The
This,
purple one?
yeah, it's purple one. So
Yeah.
So both these are nucleolus, but overall structure is nucleus, right? And that's why it
Mhm.
is labeled A, it's A. So there's no difference. Both are A structures, both are nucleus, right? Okay, then after I can get back to where's E? Yeah, here's E. And what is
Uh,
this structure? Like this structure. Like this, Filza.
it looks like cytoplasm, but it could be
Uh,
mitochondria.
mhm. Yes, it is mitochondria, but how it looks like the cytoplasm? Cytoplasm is just the medium, is just the surface, is just the place. This all structure is cytoplasm. This, all, this, all, this, all, this, all, this, all. You know, and it contains water in it, right? You just consider it's a pond, and in which all different structures are swimming, right?
Mhm.
They are placed there. So cytoplasm is actually a semi-fluid structure, a semi-fluid component, which is the part of cell, and all different subcellular structures are embedded in it. Okay, so here this all is cytoplasm that contains different structures. We'll talk about it as well. So yes, you're right. You're absolutely right here that this structure that you have identified, it's mitochondria. Okay?
Okay.
It's mitochondria. Mitochondria. Good. Okay, E, F, F, F is F. Okay, this one is F. Let's try to figure out and identify which structure it is. F. I'm talking about F now. What do you think what this structure is? Yes, that's a challenge, Phil. Again, guess. I just
Which
want
one
guess.
again?
I'm talking about this F, Filza.
F.
This one,
Uh, um. Uh,
this structure.
no, no, I don't know about this one.
I just want guess. Any guess. Whether right or wrong, I'm not going for that. I'm just saying, give one guess. Yes. Hurry up. Any
Uh,
name of any subcellular structure or organelle in the cell. You name it. But definitely that should not be that we have already labeled. So definitely two structures are not present there. So um, other than what we have labeled, you can name any of the structure. Yes, name it, name it, name it.
mm,
Okay, should I give you, I mean, hint, I mean, suppose, is this cell membrane? No, cell membrane is already labeled. Is this nucleus? No, nucleus is already labeled. Is this vacuole? Is this,
Golgi apparatus?
sorry?
Golgi apparatus. I forgot how it's pronounced.
Golgi apparatus.
Yeah.
How your guess is like 100%? I'm thinking
Because
about.
I studied yesterday.
All right. So I was just thinking like, how your guess, like, that's a good thing. Yes, it's a Golgi apparatus. Good. So your guesses are going good. That's a good news as well. Okay, now G. Phil, let's talk about this one now. Another guess.
One. Which one again?
I'm just a moment. Just a moment. Just a moment. Just a moment. Like this one. G. This one. F that we have done. It's Golgi apparatus. What's about G now?
G, 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 G. No,
Yes.
you would have to help with that one.
I just want another. Okay, I'm just going to give you a hint here. Help in the sense to give you hints. How I can give you hint? Like. We can talk about it. What is that? Yeah, I can give you an help here. Is this mitochondria? Is this mitochondrion? Is this endoplasmic reticulum? Is this Golgi apparatus? Is this chloroplast? Is this flagellum? Is this cell wall? What it is?
Nah. It's not cell wall because the wall is there.
Yeah, it's not cell wall. We have already labeled that. It's not mitochondrion. I think we have labeled that. It's not chloroplast because we have labeled it already. It's not flagellum because flagellum is something that's outside of the cell. But what's in this now? Like, is this endoplasmic reticulum?
Plasmic reticulum, no,
Why? Is this vacuole?
no.
Okay, then what is this? Like, if this is not endoplasmic reticulum, then what it is? I think it's endoplasmic reticulum as per my guess. I just want your opinion now. Why you not agree with this? Because um, endoplasmic reticulum, there are two types of it. Rough endoplasmic reticulum, smooth endoplasmic reticulum. And the rough endoplasmic reticulum is just the close vicinity of the nucleus, because once the messenger RNA will come out, rough endoplasmic reticulum contain ribosomes, and those ribosomes will just read the message that will come from the nucleus and to convert that into the protein. Oh, yeah. It's a function of the endoplasmic reticulum. So I think it should be the endoplasmic reticulum. Why you not agree with this, Filza?
Because it doesn't look like that.
Okay, just a moment. I'm going to show you. That's a good thing. This discussion is quite helpful. Endoplasmic reticulum. All right. So, just a moment. I'm copying image. Like, look at this. Definitely, this is not zoom in structure. You can see here. Is this visible to you, Filza?
Yes, it is.
So, can you identify where is rough endoplasmic reticulum, where is smooth endoplasmic reticulum? And is this not very similar to the structure which we can see here? Like this one. Okay, I'm going to show you. I'm just talking about this animal cell now. And we are also going to suppose this structure, and this structure, this one, this one. Is this not very close to this? And this one is, it's a nucleus. It's a nuclear envelope. It's nuclear pore. It's like you can see here. And this portion here it contains ribosomes. It's rough endoplasmic reticulum. So here you can see it's a smooth endoplasmic reticulum. Are you getting
Okay.
something or not? So
Yes.
now you can compare the structure with the structure that you have in the diagram. So are you agree with this? Like this is endoplasmic reticulum. We are not going to divide it into rough or smooth because this is not zoom in structure. So we don't need to mess up with this. I'm just going to give you a general name, right? And what's that? Yeah,
For
yeah.
rough endoplasmic rectum then, or ribosomes?
Rough. No, we are not going for rough or smooth. Just we would say endoplasmic reticulum. Because once you will zoom in the structure, so you will be able to see, okay, this is rough endoplasmic reticulum, as you can see here in this case. And this one is a smooth endoplasmic reticulum. And why we call it rough endoplasmic reticulum? Just because of the presence of ribosomes. Are you getting?
Yes.
So where are ribosomes? Just identify them. Hurry up.
Ribosomes. The little dots.
Yes, good. So that's our ribosomes. So where's confusion? I don't think so there is confusion. Okay, let's try to label it now. Endoplasmic reticulum. Reticulum, okay? Reticulum. So endoplasmic reticulum, right? And you know how you will identify this once you will see in the cell? Just any structure that's close to the nucleus. And this structure is quite similar to this one. Is this not?
Yes, it is.
But we call it Golgi apparatus. We didn't say like it's endoplasmic reticulum. How you will identify it's endoplasmic reticulum? Once you will see that is very close to the nucleus. that should be
Okay.
very close to the nucleus, right? And that structure, the similar structure that's away from the nucleus, you would say that's a Golgi apparatus. Both have the same vesicles and a vesicular structure. Like here we can see like this, these bags sort of structure. Okay, getting
Yeah. Yeah.
Filza? Great.
I am.
Okay, good. So now let's try to label what? Like this H. Okay, what is this H? It's a elongated tail sort of structure. It's a tail. Um, animal cell has some tail. A few of them.
That's how I recognized that it was an animal cell because it has a tail and then B is green. So green can also be identified as like plants because plants are also green. But then animals, they also have a tail. So that's how my guess was.
Okay, so let me tell you one thing. Animal cells, many of the animal cells, they have locomotory organ. Just like we have legs. We are moving by of legs. But there are some animal cells, they have some locomotory organ, just like us. We have legs. But they don't have legs. Definitely they can't be like to have legs, right? So, but there's some alternative structure there. You know what's that? Let me tell you. Flagellum. I'm writing outside here, Filza, like this.
Okay.
Can you see here? Flagellum.
Yes.
There's another structure that's called cilia. Have you ever heard this word, this term, cilia?
Celia, no.
Okay. Have you ever heard this flagellum?
Flagellum, yes, I feel like I've heard about it.
Yes. So this is a locomotory structure, not in all animal cells, remember, but this is the general structure that we have included here. This tail sort of structure. We can't say it's a tail. It's elongated fibrous structure. That's what we called flagellum, right? Cilia and flagellum both are locomotory organs in cells, but the difference is its length is bit long like this, and the cilia are small in length like this. And this is what you will see in the small intestine where it increases the surface area, and that's we'll talk about it. Okay, so this is flagellum. Let's try to label it now, Filza. Flagellum. Okay, got it? What's its
Yeah.
function? Its function is to help in locomotion, in movement of the cells. Cells can move, this cell can move from one place to another just by using this flagellum. Got it?
Yeah.
No, are you sleepy? I think you're tired because you were on the job.
No, I was in university today.
Oh, today you were in university. Yeah, okay.
Yep.
So you didn't find time to take rest, but
No, I didn't. That's
oh,
why I was late to class as well.
oh God. All right. That's really, I really appreciate like you have a good stamina to work. That's wonderful. And okay, it's okay. Now it's time to like it would be there 8 o'clock or no, it could be 8 or 7 o'clock. 7 o'clock, I think. Seven. Yeah, right? Is this the time like you prefer there? It's 7 o'clock? Seven, eight, nine. Okay. Okay, Filza. So we have here one more structure. It's G. Oh, it's similar to this. So now it's your turn to label it. Please, Filza.
Yeah.
What is the structure? I'm talking about this plant side. G is
The
here and G is
mm-hmm.
also here. What's the structure now? Can you identify the structure that's very close to the nucleus? Like this structure. Yes, Filza.
The
Guess,
G
guess,
G
guess.
is what?
Hmm. It's in plant now.
But didn't I already answer this one?
Where? But that was in
No, but
animals.
this is the thing. So if animal has it, plant has it, then this means it's the endoplasmic reticulum because both are the same shape and both are close to the nucleus.
Yes, that's great. That's I was trying to get the answer from you. That's great. So it's endoplasmic reticulum. Endoplasmic plasmic reticulum. Okay, got it? So yeah,
Yeah.
both are same structures. I mean, many of the structures which are similar in both cells. I mean, animal cell and plant cell is not much different from each other. You will see very few differences. Okay, can you see something here? Like the nucleus is in the center here, but in this case, nucleus is in the periphery. Oh God, why it is so? Let's try to understand this. What is the structure? But they didn't label it. I think we should label. Oh no no no, they have labeled. What's this D now? I'm going to write D on this side now. Okay, what is this D?
D,
It's
hold
a
up.
structure that. Can you see this the same structure in case of animal cell? I think no.
No, no.
No. It means like this is a unique structure which is only present in Filzain.
Animal cell. I mean,
This is
plant
this
cell.
yeah, plant cell, not in animal cell. Like this is the plant cell. Okay, so what this structure would be? Should I give you some? Okay, just guess it, Filza. Then after I'll give you some hints as well. Okay, guess one subcellular structure that is only present in plant cell. Yes.
I'm not gonna guess this one because I don't know.
Okay, it's okay. It's a vacuole.
Okay,
It's a vacuole. This D,
vacuole. Okay.
vacuole. And this is only present in case of yeah, plant cell.
Mm, plant cell,
Yeah.
because it's only seen in plant, not in
Yes.
animal.
And this is quite large. Can you see it? Like it's a large in size as compared to other subcellular structures. Is this not large? Is this not larger as compared to other structures that we have? Like it's bigger than even nucleus as well. Oh God, how it is. Like it's bigger than chloroplast, it's bigger than endoplasmic reticulum, it's even bigger than the mitochondria. So that's why it is there to occupy major portion of plant cell. That's why this nucleus is on the periphery right now. The nucleus position is at the periphery because of the presence of larger size vacuole in plant cell. Okay,
Mm-hmm.
got it? So it's a
Yeah.
vacuole. It stores water, it stores minerals and some specific proteins. So it is stored in the vacuole. Once it is required in the body, so that will be utilized. Okay,
Okay.
we'll talk about this individually as well, but right now we are just trying to identify it. I'm not talking about its function, but I'm just giving you a hint of this. All right.
Okay.
Yes, now we have here this I. Okay, let's try to label it. I. Yes, Filza,
Yes.
guess the structure which is not present in animal cell and it is outside of the cell as well. I'm talking about this one.
H, right?
Oh, no, 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 no. I'm talking about this one.
Mhm. This looks like a wall to me.
Yes, name it properly. Is a cell wall?
It is a cell wall.
Yes. And where's the cell wall in case of animal? Do we have?
No, there is no cell wall because the shape of it is very circular.
Yes, good. So there's no cell wall in case of animal cell. So this is the big difference. Keep in mind, animal cell never contains cell wall, but it contains cell membrane. Cell membrane is also present in plant cell, but there's additional structure. Its name is cell wall. Okay, getting?
Yeah.
All right. So what we can do now? Yeah, done from. Is there any structure that? Oh yeah. I think we have done about this. This this edge. Edge is a flagellum. Any structure which is left? Yeah, this F. What is this F?
Which one, F?
On this plant cell. F in plant cell.
I'm gonna say Golgi apparatus.
Sorry?
Uh, I can't
Golgi
pronounce.
apparatus. Golgi. It is pronounced in two ways. Golgi apparatus or Golgi apparatus. In Canada,
Golgi apparatus. Okay.
yeah, you can pronounce it Golgi apparatus. Okay?
Okay.
Done. Great. Appreciate yourself. I mean, yeah, because this is what all the differences that we have in animal cell and plant cell. And you must be able to identify this before going to start photosynthesis and respiration. Okay,
Okay.
got it?
But I'm gonna end the class at 6 because what is it called? I'm not feeling well actually. I do have a fever.
Oh, oh my goodness. Okay. All right, Filza. Yeah, I can understand. I can understand. I can understand. You can, I mean, whenever you feel good, you can suppose if I have free class, so I can give you a makeover, no worries.
Sure,
Um,
that would be great.
sorry?
That would be great.
Yeah, yeah. I'm already always available for you. But suppose there should not be a class at that point of time. You should confirm this for me as well, or you can to the management. So there's no, I mean, issue with me to give you a class. Okay, take a moment, 2 minutes break. Then after last 10 minutes, we'll talk about one more concept, and I hope you are good in that. And just for 2 minutes, you can take a deep breath.
Sure.
Okay, done. Okay,
Yes.
great, Filza. So let's talk about one more concept because in order to start photosynthesis and respiration, we must have one more concept. Let's talk about that. What?
Okay.
Yeah, that concept is I'm going to paste a very good picture in front of you. Filza, here it is. Look at this, Buddha. Can you see? Is this visible
Yes.
to you?
Isn't this solubility or osmosis?
Yeah. Like we are going to talk
Osmosis.
about. I mean, just try to look into the first three beaker, like the figure two. Can you see here something? Like this one, this one, this one. What's the difference? Try to understand what's going on here. Well, I'm also trying to understand, Filza. Let's try to focus on this now. Suppose, consider one thing, Filza. Suppose you have a glass of water.
Yeah.
you are going to put a drop of ink or any coloring substance into that. Your liquid coloring substance, that would be ink, because that's more familiar to us. Let's try to put in that. This drop is going to be spreaded. Like this graph is spreaded. Once it was just at this point, then now it's getting like this, like this. Can you see it? And then in the last beaker, you can see this is spreaded in all over this solution. What's going on here?
It's
What
dissolving, like the particles are.
do you know about diffusion? What is diffusion? The diffusion is the process in which the molecules are moving from higher concentration to lower concentration. You know, this drop which is placed here, I mean, once you put into that beaker, that was actually half filled of the water. So, but it was less in concentration here in this area, but it is more in concentration in this area. So this ink is going to move from higher concentration, I mean, from higher gradient to lower gradient, from higher concentration gradient to lower concentration gradient. So it's going on like this. It's spreaded, spreaded, it's spreaded. So once you'll see, this is now going to be uniform in all of the speaker. There's no further diffusion. I mean, that ink is going to be spreaded throughout that beaker. And you can experience this from yourself. You can take a glass, you can take a bigger or transparent glass. You can put a drop of ink in that. You will see after some time that ink would be just like this. That ink will be spreaded in all of that beaker. But it would started like this. You will see at the first stage, you will see like this. Then after some time, you will see like this. Then after some time, suppose it would be after 10 minutes, you will see this sort of appearance of that glass. Okay?
Okay.
This is diffusion, Filza.
Diffusion, the first one, right?
All of this, like I'm talking
All of
about,
them?
I'm talking about this figure two. I'm just talking about the figure two for you. You know, this all over the figure two, you will see this is diffusion, this is diffusion, this, I mean, this is just the concentration. I mean, once this was the initial stage, this is second stage, this is third stage. All this is diffusion in this figure two. Like it is actually showing you how this is going on. The movement of molecules from higher concentration to lower concentration. Filza, are you confused
Yeah.
somewhere? Please ask
Uh,
question.
no. So these three are diffusion.
Yes, and you know, I mean, this is the start of the diffusion. Like, suppose, let me tell you one thing. Suppose you are right now. You are in any room, right? You are somewhere in the room
Yes.
or outside. Okay.
No,
So
yeah.
if anybody comes at your door and spray a perfume,
Mm-hmm.
a room sprayer, right? So you know you are sitting bit far away from that door, and definitely at the very early stage you will not feel that fragrance, but after some time you will see the molecules will flow in that air and they will reach to you as well, and you will feel okay, this is the fragrance of the any room spray or room freshener, or that's a perfume, whatever it is. So why you didn't feel at the very early stage, at the very start?
Could you
Why?
repeat that again?
What I'm saying, you are sitting bit away from the door. Somebody
Yeah.
comes, your any of the siblings, like your brother's coming, and he just spray a room freshener or any perfume at the door.
It's going to diffuse in the room. It's going to spread out.
Yes. Once it will reach to you, actually this is spread out in that room, right? But after some time, it will take time. Once it was okay. If you'll just get up from there and to move towards the door, you can feel that fragrance easily, more early. But because you are sitting bit far away from the door, so that's why that spread is going to take some time, right? So this is because of the diffusion, because the molecules were higher in concentration at door and they're going towards the lower concentration towards you. Because at your point, concentration is 0, but higher concentration of that room freshener or that perfume is at the door. So it will just float in the air and they will reach up to you. That's a diffusion. This process is called diffusion. Getting?
Yes.
And
So it
it's
spreads out. Basically, when it is given in an amount, it will spread out itself.
yes, that's it. And that's yes, there's no need to provide any energy. Are your brothers providing some energy there? No, he's just praying. He's just okay. All right. Are you getting this point, Filza, or not?
Yes, I am.
Sorry. Okay, that's fine. So I hope you're getting okay.
Okay.
Let's one more point, Filza. We have 3 minutes, so I'm just going to conclude for you. And can you see here in figure 3 what's going on? Let's try to understand. Here's the water, okay? And
Okay.
there's a membrane here. This is the membrane. This is membrane. Can you see here? This is the membrane. These are
Yes.
molecules. And these molecules, once they were greater in concentration here, and they're going to move towards other side of it. Like you have 4 molecules here. This is before. Okay, what's going on? You know what? This is one case. This A is concentration gradient. It is built across the membrane. Can you see here in the figure 2 after what's going on?
Figure 2.
You will tell me.
So it was divided into two, right? Some
I'm
particles went the other
listen,
side.
listen, Filza. I'm going to ask you
Uh-huh.
some questions. Filza, one more point. Then after you conclude, please, because you are going to somewhere. You're not
Yeah.
feeling well. So I'm not going to decide to put this arrow. Can you please tell me on which direction this water has to move? This direction or this direction?
The
I'm
other direction.
I'm in this one. This one.
Yes. Yes.
Can you please tell me why?
Um,
Water concentration. Please find out the water concentration. Water is more here as compared to this.
yeah.
Water concentration is more as compared to these molecules which are here. These are only few. So more water is present in this side. So what do you think? Water should come from, suppose I'm saying this is C, this is D. So water should come from C to D or D to C?
C to uh D to C.
Yes, good, good. So you know about this. Like because the water concentration is more on D side as compared to C side, because on C side we can see there are number of other molecules. So concentration is greater. So we can say water should move from D to C. That's why you can see here. But remember, Filza, it's crossing the cell membrane. It's crossing the membrane or the barrier. So you can see here now what is getting down here and what is getting increased here. This is the condition after, and this is the condition before. You know what's going on here? Can you name the process which is there?
No,
I'm
I
going to
cannot.
listen. The movement of water molecules from higher concentration to lower concentration across the semi-permeable membrane. What is this? Can you name the what I have just defined it? Can you name the term?
The for
Osmosis.
osmosis. So this is osmosis.
Yes, the movement of molecules from higher concentration, the movement of the water molecules, remember, the movement of the water molecules from higher concentration to lower concentration through semi-permeable membrane. Is this not semi-permeable membrane, Filza?
It is.
Yes, and that's what we call osmosis. Have you applied any source of energy here?
No.
No, it's a passive process. It's not active process, it's a passive process, right? Remember.
Yes.
So there's no need to provide energy because what is already there's a gradient, concentration gradient is here. So what is moving from high concentration to lower concentration? Getting?
Yeah.
So we have covered here two concepts like diffusion and osmosis, right? So this is what we should know. And before that, we have just tried to understand, identify different structures which are present in plant cell and animal cell. Got it?
Yes.
So let's conclude this discussion for today. As you are not feeling well, so otherwise, my plan was to start chemistry on today. It's okay. We'll study chemistry in next week first, and then we'll go for the biology. Okay. This is how we can proceed next week. All right.
Yeah.
Okay. Let's conclude this for today. Take care of yourself. Bye bye. See you
Thank you. You
next
too.
week.
Bye.
Take care.
Bye.
Bye.